Welcome to Success Blueprint for JEE and NEET. Join the class 11th Biology.
Study at your own pace and save your time and energy. Please like and subscribe for more educational videos.
Introduction.
Good morning, teacher.
Good morning, students. Today, we will learn biomolecules. Teacher, what are the biomolecules? A biomolecule is an organic molecule that is produced by a living organism. Teacher, the molecules produced in our body are biomolecules. Yes, Ram. Can anybody give me some examples of biomolecules?
Teacher, there are many examples such as proteins, polysaccharides, nucleic acids, etc.
Very good, Shweta. In this lesson, you will learn biomolecules in animals. Objectives. At the end of this lesson, you will be able to analyze chemical composition, explain nucleotides, discuss primary and secondary metabolites, know biomacromolecules, discuss proteins, discuss polysaccharides, discuss nucleic acids, describe structure of proteins, discuss nature of bond-linking monomers in a polymer, discuss dynamic state of body constituent, concept of metabolism, explain metabolic basis for living, explain the living state, describe enzymes, how to analyze chemical composition. Biomolecules are those molecules that are involved in the maintenance and metabolic processes of living organisms. Most of the chemical compounds present in living organisms always contain carbon and hydrogen, and their carbon atoms are covalently bonded to form straight chain, branched chain, or ring structures. Organic compounds can be classified into amino acids, nucleotides, bases, fatty acids, etc. Amino acids. Amino acids act as a building blocks of protein and intermediates in metabolism. Amino acids are molecules that have amino group, a carboxylic acid group, a hydrogen, and a characteristic side chain all attached one carbon atom. The most common amino acids are the alpha amino acids. The side chains or R groups can range from a hydrogen atom to a large complicated side chains. The physical and chemical properties unique to each amino acids are the result of the structure and chemical properties of the R group. On the basis of number of carboxyl and amino groups, there are acidic, basic and neutral amino acids. Glutamic acid is acidic amino acids. Lysine is basic amino acids. Valine is neutral amino acids. Lipids. Lipids are organic molecules that are insoluble in water. Three examples of lipids are neutral fats, phospholipids and cholesterol. Neutral fats. Three fatty acid chains attached to a single glycerol molecule by dehydration synthesis. Neutral fat stores energy fuel, insulates body tissues and protects organs. Phospholipids. Phospholipids have a non-polar fatty acid chain portion and a phosphate polar portion. Two layers of phospholipids form the chief component of all cell membranes. Two layers of phospholipid molecules self-assemble so that their hydrophilic heads form the surface and interior of the membrane, and the hydrophobic tails face each other. Cholesterol. The cholesterol molecule has 4 interconnected carbon rings. Nucleotides. Nucleotide is a basic building block of nucleic acids such as DNA and RNA. It is an organic compound made up of nitrogenous base, a sugar, and a phosphate group. Some of the nitrogen bases are adenine, thymine, guanine, cytosine, and uracil. If the nitrogen bases are attached to sugar, then they are called as nucleosides. Adenosine, guanosine, thymidine, uridine, and cytidine are nucleosides. Adenylic acid, thymidyllic acid, guanylic acid, uridylic acid, and cytidyllic acid are nucleotides. Primary and secondary metabolites. A primary metabolite is metabolite that is directly involved in normal growth, development, and reproduction. It includes different types of organic compounds. Example, sucrose and starch, cellulose, DNA, RNA, pigments such as chlorophyll. A secondary metabolite is involved in specific functions such as pollinator attraction or defense against herbivore. They can be classified into three categories: alkaloids, terpenoids, phenolics. Alkaloids are a large group of nitrogen-containing compounds. Terpenoids are derived from acetyl coenzyme A or from intermediates in glycolysis. Phenolic compounds are defined by the presence of one or more aromatic rings bearing a hydroxyl functional group. Biomacromolecules. Compounds found in acid-soluble pool, and they have a molecular weight ranging from 18 to 800 Dalton approximately. The biomacromolecules which fall in the acid-insoluble fraction include proteins, nucleic acids, polysaccharides, and lipids. All biomacromolecules have molecular weight more than 10,000 Da, except lipids. This is because, except lipids, all are polymeric substances. Polysaccharides. Polysaccharides are the polymers of monosaccharides. Polysaccharides can be homopolymer or heteropolymer of monosaccharides. Nucleic acid. Nucleic acids are the polymers of nucleotides. Proteins. Proteins are polymers of amino acids. Proteins are polypeptides where amino acids are linked by peptide bonds in the form of a linear chain. Functions of proteins. Transportation of nutrients across membranes, fight infections, and act as hormones and enzymes. Proteins. Proteins are polymers of amino acids, covalently linked through peptide bonds into a chain. Proteins act as antibodies involved in defending body from antigens. Contractile protein is responsible for movement. For example, actin and myosin. Proteins are polymers of amino acids joined together by peptide bonds. There are 21 types of amino acids that make every protein. Each of the amino acid has a fundamental design composed of central carbon bonded to a hydrogen, a carboxyl group, an amino group, and a unique side chain or R group. Polysaccharides. Polysaccharides are the complex carbohydrates. They are made up of chains of monosaccharides which are bonded together by glycosidic bonds. Cellulose is a major component of plant cell walls. It is an unbranched polymer with about 10,000 glucose units per chain. Chitin is an unbranched polymer of N-acetyl D-glucosamine. It is found in fungi and is the main component of arthropod and lower animal exoskeletons, example, insect, crab, and shrimp shells. Glycogen is a polysaccharide composed of glucose units linked by alpha-1,4 glycosidic bonds. with alpha 1,6 glycosidic bonds, which provides branching points. Assessment. Before proceeding further, let us know how much have you learned. Drag and drop the correct option. Nucleic acids. Nucleotides are the building blocks of all nucleic acids. Nucleotides have a distinctive structure composed of three components covalently bound together. A nitrogen-containing base, either a pyrimidine or purine. A 5-carbon sugar, ribose or deoxyribose. A phosphate group. The combination of a base and a sugar is called a nucleoside. Structure of proteins. Proteins consists of one or more chains of amino acids linked by peptide bonds and are folded into a specific three-dimensional shape. An amino acid is composed of central carbon atom bonded to a hydrogen, a carboxyl group, an amino group, and a unique side chain or R group. Levels of protein structure. Primary structure. The linear settlement of amino acids in a protein and the location of covalent linkages such as disulfide bonds in between amino acids. Secondary structure. Areas of folding within a protein. Example include alpha helices and pleated sheets, which are stabilized by hydrogen bonding. Tertiary structure. The final three-dimensional structure of a protein, which results from a large number of non-covalent interactions between amino acids. Quaternary structure. Non-covalent interactions that bind multiple polypeptides into a single larger protein. Hemoglobin has quaternary structure due to association of 2-alpha-globin and 2-beta-globin polyproteins. Nature of bond-linking monomers in a polymer. Peptide bond. This bond is formed when the carboxyl group of one amino acid reacts with the amino group of the next amino acid with the removal of water molecule. It links the amino acids in a polypeptide chain. Glycosidic bond. This bond links two carbon atoms of adjacent monosaccharides to form polysaccharides. It also involves dehydration. Phosphodiester bond. This bond links two nucleotides to form nucleic acid. It is formed between the phosphate and hydroxyl groups of sugar. Dynamic state of body constituents. Concept of metabolism. Metabolism is a process involving a set of chemical reactions that changes a biomolecule into another, either for storage, another reaction, or as a byproduct. Example of anabolic reactions: the formation of glycogen from glucose, the formation of proteins from amino acids, the formation of storage fat from fatty acids. Metabolic basis for living. The metabolic breakdown of complex molecules into simpler molecules leads to release of energy is catabolic reactions. Examples of catabolic reactions: glycolysis. It is responsible for the production of adenosine triphosphate through the degradation of glucose. Glycogenolysis. Hydrolysis of glycogen occurs in muscle cells for production of energy and in liver cells for maintaining blood sugar level. The living state. The most important fact of biological system is that living organisms exist in a steady state characterized by concentrations of each biomolecule. The chemical or physical process moves spontaneously to equilibrium, and the steady state is the non-equilibrium state. All living organisms work continuously. They cannot reach to equilibrium, hence the living state is a non-equilibrium steady state to be able to perform work. living process is a constant effort to prevent falling into equilibrium. It could be achieved by energy input. Through metabolism, energy is produced. Hence, the living state and metabolism are same. Without metabolism, there cannot be a living state. Enzymes. An enzyme is a substance which is produced by a living organism, act as a catalyst to bring about a biochemical reaction. Almost all enzymes are proteins. Enzymes also have primary, secondary, and tertiary structures like all proteins. At the active site, the substrate fits in and reaction is catalyzed at a high rate. Chemical reactions. Chemical compound exhibit two types of changes. Physical change means change in shape without breaking of bonds, and chemical change involves breaking of bonds. The rate of physical and chemical reactions can be measured as the amount of product formed per unit time. Rate of reaction delta P by delta T. Temperature directly affects the rate of reaction. Rate doubles or halves for every 10 degrees Celsius changes in either direction. Due to increase in absolute temperature, the average kinetic energy increases and the number of molecules with energy greater than the threshold energy also increases. Therefore, it is observed that the rate of reaction increases with increase in temperature. An enzyme can only catalyze a specific chemical reaction. For example, the sugar maltose is made of two glucose molecules bonded together. The enzyme maltase is shaped in such a way that it can break the bond and free the two glucose molecules. How do enzymes bring about such high rates of chemical conversions? The enzyme's ability to increase the reaction rate depends on the fact that it stabilizes the transition state. The energy in transition state is activation energy, which is defined as the minimum energy that is needed to break certain bonds of the reactants to convert them into product. Enzymes decreases activation energy by shaping its active site such that it fits the transition state even better than the substrate. For example, one molecule of catalase can break 40 million molecules of hydrogen peroxide each second. Nature of enzyme action. Catalytic cycle of enzyme action. The substrate binds to the active site of enzyme. The active site is the specific region of the enzyme which combines with the substrate. The enzyme changes the shape of the substrate so that substrate fits more tightly. The active site of the enzyme breaks the bond of the substrate and new enzyme product complex is formed. The enzyme releases the product and the enzyme is ready to bind another molecule. Factors affecting enzyme activity. Temperature and pH. The activity of enzymes is strongly affected by changes in pH and temperature. Each enzyme works best at a certain pH and temperature. Its activity decreases at values above and below that point. For example, the protease pepsin works best at a pH of 1 to 2. The protease trypsin is inactive at such a low pH but very active at a pH of 8. Changes in pH alter the state of ionization of charged amino acids that may play a crucial role in substrate binding action. Concentration of substrate. If the amount of enzyme is kept constant and the substrate concentration is then increased step by step, the reaction velocity will increase until it reaches a maximum. After that point, increase in substrate concentration will not increase the velocity. Inhibitors are substances which change the catalytic action of the enzyme and consequently slow down or stop catalysis. Competitive inhibition occurs when the substrate and a substance resembling the substrate are both added to the enzyme. Classification and nomenclature of enzymes. The enzymes can be classified into different groups based on the type of reactions. Oxidoreductases or dehydrogenases. The enzymes that transfer electron from one molecule to another molecule are called oxidoreductase. Most of the metabolic oxidation reactions involve removing hydrogen from the electron donor. These enzymes are called dehydrogenases. Transferases. These are the enzymes that involves in transferring a group. Hydrolases. An enzyme that speeds up the process of hydrolysis. Classification and nomenclature of enzymes. Lysases. These are the enzymes that catalyze the cleavage of CC, CO and other bonds by means other than by hydrolysis or oxidation. Isomerases. These enzymes catalyze the structural rearrangement of isomers, for example, fumarase. Ligases. These enzymes join two molecules by the formation of new bonds, for example, citric acid. Cofactors. Cofactors are defined as molecules that closely associate with enzymes during enzymatic reactions and are converted from one form to another in the reaction. Three kinds of cofactors are prosthetic groups, coenzymes, and metal ions. Prosthetic group. A tightly bounded non-polypeptide structure required for the activity of an enzyme. For example, the heme of hemoglobin. Cofactors. Coenzymes. These are the organic cofactors such as nicotinamide adenine dinucleotide, that is NAD, and flavin mononucleotide, that is FMN, are involved in oxidation reduction reactions. Metal ions. Metal ions participate in oxidation reduction reactions, stabilize charge complexes in enzymes. Assessment. Before proceeding further, let us know how much have you learned. Match the following. Did you know? Watching horror movies can increase the metabolic rate. Cinnamon is the spice that can increase your metabolism rate. If you skip meals, it can slow down your metabolism. Summary. Let us summarize what we have learned. Biomolecules are those molecules that are involved in the maintenance and metabolic processes of living organisms. Amino acids act as building blocks of protein and intermediates in metabolism. Nucleotide is a basic building block of nucleic acids such as DNA and RNA. Polysaccharides are the polymers of monosaccharides. Nucleic acids are the polymers of nucleotides. Proteins are polymers of amino acids. The four levels of protein are primary, secondary, tertiary, quaternary structure. Metabolism is a process involving a set of chemical reactions that changes a biomolecule into another, either for storage, another reaction, or as a byproduct. Almost all enzymes are proteins. The enzyme's ability to increase the reaction rate depends on the fact that it stabilizes the transition state. Cofactors are defined as molecules that closely associate with enzyme during enzymatic reactions and are converted from one form to another in the reaction.
Friends, welcome to Kwiiz Success. Are you tired of memorizing without really understanding concepts for exams? Our innovative learning techniques help students grasp concepts easily and effectively. Our Success Blueprint course for class 11 and 12 gives you a real exam-like experience, helping you build confidence and score higher in IITG and NEET. Here, you'll find the perfect combination of 3D animated videos, AI-powered test series, and ebooks enriched with MCQs and mind maps. All our courses are designed by IIT and IIM alumni to ensure top-quality learning. For online courses and ebooks, visit www.success.org.in. Download the Success Kwiiz mobile app for free. For advanced test series and free mock exams, visit www.kwiiz.success.org.in. Download the Kwiiz Success mobile app and get a real improvement in your scores. Success is now in your hands. Thousands of students have already benefited from Kwiiz Success. Now it's your turn. Study from home, learn at your own pace, and prepare at your convenience. Don't forget to subscribe to our YouTube channel, Kwiiz Success, and share it with your friends. Thank you, and all the best for your success.